Meiosis, the cellular division of gametes, consists of two stages, meiosis 1 and meiosis 2, that divides a cell twice to create 4 haploid cells for reproduction. In a phase of meiosis, the germ cell duplicates its diploid set of uncondensed chromosomes. The two duplicated centrosomes of the cell begin in the center of the cell and move towards the polar ends to generate the meiotic spindle apparatus. In prophase 1, the longest stage of meiosis, the nuclear envelope breaks down. The chromatin now condenses, and sister chromatids connected by a centromere form. In this phase, the chromosomes cross over as the tetrads interact with their homologous pair at their chiasmatas. Only the sister chromatids connected at the chiasmatas exchange genetic material. This process creates recombinant chromosomes that contain different genetic material from each parent. Like in mitosis, the microtubules from the centrosomes attach to the kinetochores, which are specialized protein structures at the centromere of the homolog pairs as they prepare for metaphase. Driven by the motor proteins in the kinetochores, the homolog pairs line up at the metaphase plate, each pair facing opposite ends of the cell as metaphase 1 begins. The random assortment of chromosomes along the metaphase plate accounts for the law of independent assortment, which is the notion that the alleles from each parent are randomly distributed into the gametes. In anaphase 1, the cohesion of the proteins in the centromere break down, which allow the homologs to separate. The separation of the homologous pairs is evidence of the law of segregation, which accounts for the inheritance of one allele for each gene. Anaphase 1 demonstrates this law as the homologous pair because the duplicated chromosomes for the same trait are moved to the other poles of the cell because of the guiding of the spindle apparatus, the complex of microtubules controlled by the centrosomes. Telophase 1 begins with a haploid amount of duplicated sister chromatids on either pole of the cell. At this time, the cleavage furrow for cytokinesis begins to form, which is the splitting of the cytoplasm and separation of the two haploid sets of duplicated chromosomes into two haploid daughter cells. At this time, some animal species form nuclear envelopes around the chromosomes. From meiosis 1 to meiosis 2, no further replication of the chromosomes occur. Meiosis 2. Prophase 2 begins with two daughter cells, and the spindle apparatus from the now duplicated centrosomes emerge as the chromosomes in each cell travel towards the metaphase plate. In some species, the nuclear envelope fragments once more.
You guys know where Mr. Strang is?
In metaphase 2, once positioned on the metaphase plate from prophase 2, the duplicated chromosomes composed of non-genetically identical chromatids attached to the kinetochores of the microtubules of the spindle apparatus. In anaphase 2, the sister chromatids separate at the centromeres due to the breakdown of proteins between them. This causes the separation of the duplicated chromosomes as the chromatids move towards opposite poles as individual chromosomes. The spindle apparatus and kinetochores facilitate the separation. At the beginning of telophase 2, the polar ends of each cell contain unduplicated haploid amounts of chromosomes. At this time, the cleavage furrow of the cell wall is created, and 4 daughter cells are generated due to the cytokinesis or the separation of cytoplasm. These 4 haploid daughter cells contain genetically distinct DNA, each with a haploid set of unduplicated chromosomes. Their chromatin also decondenses and the nuclear envelope forms. In male animals, 4 sperm cells form, and in female animals, only 1 egg cell remains after going through the process of oogenesis.